So welcome to the 2015 NASA Ames Summer Series. Space exploration allows us to investigate the frontiers of space, our future. It also allows us to make science fiction a reality. And in the process, we'll learn about ourselves and our home planet, Earth. One of the ways that we study and investigate space and make sure that we survive that environment is to use model organisms. NASA Ames is the lead research center for conducting, managing, and building hardware to conduct rodent research in space. Today's seminar, entitled Flying Through the Ages, Rodent Research for Human Health, will be given by Dr. Ruth Globus. Ruth earned a BA degree in sociology in 1979 from the University of California at Santa Cruz, followed by another BA degree in 1981 in biology from the same university. After that, she worked at the lab of Emily Holton here at NASA Ames, where she got introduced and got the bug of being at Ames Research Center. She worked for 2 years, and then she realized that her future lies in being a PI and doing space research and ground research with rodents. So she went on to get a PhD degree in endocrinology from the University of California at San Francisco, followed by a postdoc in cell biology. She became a principal investigator at NASA Ames in 1993, and then joined the civil servant staff in 1997. She has numerous awards and publications, too many to go here at this moment. Please join me in welcoming Dr. Ruth Globus.
Thank you very much, Jacob. Thank you, Ames, for giving me the opportunity to share our work. I won't say my work, I'll say our work, and I think as I proceed through this talk, you'll see why. We have been actively engaged in developing capability to conduct long-duration rodent research on the space station. And it makes sense. Why do we want to do that? What's our big goal? Where are we going? So if we accept as our big goal, we want to have long-duration human habitation in space, then let's set as a goal to stay healthy while we do that, both during and after we come home to Earth. So ambitious goals call for ambitious questions. And here are some of the questions that came to mind as I thought about it. What are the biological changes that are relevant to human health? What changes occur, and when do they occur? How far do the adverse changes progress? Some changes may or may not have an adverse effect. So what, if anything, do we need to do about those responses? Second big question that you'll see emerging during this talk is really, how do these changes come about? What are the fundamental mechanisms at the molecular, cellular, and physiological levels that lead to observed responses? We want to do this both to better understand human biology and disease on Earth, and also we hope that will lead to a better ability to predict changes that occur and to identify interventions that may be needed. In short, these type of mechanistic studies that animal research and other analog research makes possible is to take some of the guesswork out of making decisions for our future in space. So a general outline of what I want to talk to you about today is first to provide a background. What are some of the challenges of the space environment? Why do we study rodents? I'll touch briefly on some of the past, some of the what we've learned. And then I'm going to go into some detail into what we're doing now with the Rodent Research Project, the challenges we face in accomplishing our objectives, what the capabilities are, and some of our new and surprising findings that we've obtained. So first, what are the challenges that are posed by going into a space environment? This should pose no surprise to people in this audience and thinking about the problem. We've all evolved on Earth in 1G, all species. This is continuous, except for very transient and short periods of acceleration. So our cells, our living systems, the intact organism has evolved under that influence. And in fact, we already know quite well that we can adapt to changes in that. We haven't been in that environment for much more than 2 years at the most, so there's still many unanswered questions. The second important aspect of the space environment that most are aware of is space radiation. Space radiation is unique, both in type and in exposure rates, and so it poses unique challenges, potentially both short-term and long-term, to the health of astronaut crew. But that's not all. Those aren't the only challenges we face when we go into space. Here are some of the others. Now, these might look somewhat familiar to you, because these are challenges that we certainly face on Earth as well. And in fact, things like nutrition or demanding workload we have a great deal of understanding of. Others, somewhat less. For example, in the ISS environment, there is a low but elevated level of carbon dioxide that the crew breathes in, and that has a biological effect. We know something about that from studies on long-duration submariners. But when you combine all these challenges and these factors together, you end up with a realization that it is not possible with reliability to predict the long-term consequences of these environments, especially as we seek to live in space. So what are those changes that occur? Now, in microgravity, we have widespread unloading or musculoskeletal disuse. Also, because our circulatory system has evolved in 1G, the entire system is tuned to that. And when we go into space, there's a fluid shift, more an equalization of distribution of that fluid. And many different organ systems in the body are affected and yield adaptations, some successful, some somewhat less successful. And in fact, as I've already mentioned, we understand a lot. We've been going to space for a while now. We know that there's muscle atrophy. We know that there's cardiovascular deconditioning and that there's very rapid vestibular responses. I'm heightening bone decrements here because this is my area of research and something I feel a lot of passion about. So we will talk in a little more depth about that to illustrate some of the points I'd like to make. But these points equally pertain, differing in detail, to some of the other systems that are affected by spaceflight. And in bone decrements, what do we know about fracture repair in space in humans? Nothing. It hasn't happened yet. What do we know about mechanisms? We know something, but very little. We've also flown rodents, and I'll talk a little bit about some of that history in a moment. We know things from rodent research as well, and we're making progress in getting answers to some of these really important questions from that research already. In pointing to my example here in terms of the bone decrements, already 2 spaceflight experiments have looked at the ability of bone to repair from a fracture, and in fact have found that deficits do occur. We also have more detailed understanding of mechanisms than we've been able to obtain from human flights. So when I talk about mechanisms, what do I mean here? In fact, there are a hierarchy of mechanisms that we could think about when we're trying to solve a problem. And typically in biology, we can start at the smaller level from the molecular, understanding which specific molecules are responsible for a given outcome. The molecules organize into cells, into communities of cells, into tissues, into organs, and into organ systems. And I'd like to pause here for a moment to point out, as we all know, one organ system or one tissue in our body communicates with many others. They don't exist in and of themselves to yield a healthy organism. They work together. So in the end, these together combine to result in the observed behavior and biological function of the organism, in this case, the mammal rodent. Now, you might ask me, why rodents? Why do we study rodents? The simplest answer is because of the benefits that it can be accrued and have already been shown to be accrued using rats and mice primarily. 71 of the past Nobel Prizes in medicine have been awarded to people who have used animals in their research, and it's made possible discoveries that simply wouldn't have been possible otherwise, such as the fracture healing studies that have been conducted. We don't embark lightly on a plan to study rodents in long duration habitation in space and develop the hardware and the plan and conduct the experiments. We do that under the advice of experts who review what's needed. And in the National Research Council in 2011 produced a report on really looking carefully at what's needed in both the life and physical sciences for space exploration, and point out that the lack of a facility for conducting long-term rodent research on the station is a major impediment to achieve important goals for astronaut health. And it's important to note here also that we observe carefully the federal regulations and requirements to ensure the well-being of the animals. So here's another interesting scientific reason why rodents, and we can think about this together. Aging. The typical lifespan of a human, depending on many variables, can be 70 to 90 years. Typical lifespan of a mouse is 2 years. Now, despite this enormous difference in lifespan, rodents acquire age-related diseases that very closely resemble those of humans. Osteoporosis, the loss of bone, cardiovascular deconditioning, muscle wasting. These are just several examples. So what that means is if you look at various stages during aging, you have a compressed timeline. Here you see in humans, age of 20 to 30-year-old human in years corresponds to a 3 to 6 month old animal in mice, and also approximately rats, going on to older and older ages. So what's the consequence of this for us when we're trying to solve the problem of influence of long duration habitation in space on human health? Well, let's grab a hypothesis. We'll look at the hypothesis that's really been long-standing for many years, that's been based on the observation that age-related disease such as bone loss and muscle loss is observed and it's very similar in the spaceflight environment, as I just mentioned. The hypothesis is that living in space accelerates aging. Let me emphasize, this is a hypothesis. This is not a fact. What's it going to take for us to test this hypothesis? The age of our astronauts are in the approximately 38 to 47-year range. This is a 1-year-old animal. If you want to do a lifespan experiment, let's just say, design that experiment, that would require roughly 40 years for humans. Not very practical right now. That same experiment could take about a year. Much more doable. So now I'd like to describe to you in a little more detail the biology of the changes that occur in the microgravity environment. I just spoke to you about aging, but very similar changes can occur and have been shown to occur in various experiments with aging, with radiation exposures, disuse on Earth, and hormonal changes. In humans, it takes months to decades to go from a bone that looks like this to a bone that looks like this in response to these factors. This is a micro-CT. These are both micro-CT images, microcomputed tomography images, that show the three-dimensional structure of cancellous bone, which is the highly metabolically active bone that's inside the outside shell. This looks like this in humans. It also looks like this in rodents. But we can see this depending on the stimulus, even in a matter of days. Now, how do we get to a structure that looks like this to one that looks like this? It's a product of cells. We don't think about this in the context of bone, but just like the other tissues, cells contribute to the growth and maintenance of tissue function. And I'm going to talk more about that now in the context of what we've learned from spaceflight. We would like to understand at the cellular level, as well as molecular level, which cells are responsible for the changes in bone structure that occur in the space environment. Two different lineages or derivations of cells are found in the bone marrow that give rise to the most differentiated or mature cells, the osteoclasts, which break down bone, and the osteoblasts, which build or form bone. Together, these cells define whether a bone looks like this or this. These cells derive originally from stem cells, two separate lineages, which divide and mature sequentially to become the mature cells. Where in this process do we see defects? And we've acquired information from rodent experiments that the defect is not found only in the mature cells that are responsible for breaking down and forming new bone, but also in the earlier progenitors and precursors that supply a continuous source of cells throughout life. So now that I've introduced you to some of the concepts that we'll be talking about, let's get into some details about what we've learned from spaceflight experiments and the platforms. And I'll only have time to touch on those briefly. But there have been 3 different space platforms that have been used to date. The Cosmos missions, which have grown up until recently into continued into the Bion missions, are unmanned missions by the Russians, and really in these missions performed groundbreaking work in the midst of the Cold War to determine what are the basic physiological responses of rodents and mammals to the spaceflight environment. Many more experiments were conducted in the shuttle era, and we're coming to the present in the ISS. So to summarize some of the most important features, take-home features from this shuttle era of experimentation, we really, one of the key aspects of the shuttle program was it provided the opportunity to do multiple experiments, gathering new information and taking the next logical step, which really formulates the basis of making progress in research. And in that frequent access that was made possible in that program allowed us to both define responses and test treatments. Now, what are some of the gaps in knowledge that came as a consequence of the structure of the shuttle program? One was the duration. We never learned what happens after 3 weeks. That was the longest flight. In that period, we mostly, not exclusively, but mostly studied growing rats, not adult animals. We know basic biology that the processes that control growth are very different than the processes that control maintenance or aging. So what about adults? All but 2 of the 27 rodent experiments that were conducted using the shuttle platform entailed returning the animals to Earth and recovering tissues and studying them. Now, this introduces an additional variable of reentry, landing, and a time delay, which in the shuttle era was relatively brief, only a few hours. But we know even that brief period can result in a change in outcome, depending on what variable is being analyzed. So these findings really point to the science value for doing on-orbit sample recovery. Now, moving beyond the shuttle into long-duration missions, the first one was performed on the ISS using a mouse drawer system developed by the Italians. They achieved 91 days on station, launched in 2009 via the shuttle. Male mice were flown, and samples were analyzed after returned. Now, this flight resulted in a limited number of animals recovered. Nonetheless, there are some interesting new findings that invite further study, and citations mentioned here. And as of today, we see more than 5 index papers describing this study. More recently, we've had an unmanned Bion M1 mission, which is a Russian mission. US investigators work closely with them in tissue sharing. This is a 30-day mission that flew older male animals. Again, this is a sample recovery after landing, in this case 13 hours, a fairly long period of time. And new results are still emerging from these studies. More than 13 papers have been published to date. So learning from these examples, what are our main objectives going into the Rodent Research Project here at Ames? What kind of gaps in knowledge do we want to fill, and what do we need to do to fill those? First, we knew we wanted to provide reliable, long-duration habitat for rodents on the ISS. We wanted a habitat that could support the animals in groups or individually. Rodents are social animals. They like to live together. It's important to have that capability. We also wanted the hardware to have the potential for future modifications so that we can support eventually multiple generations in space. It also needed to have relatively low maintenance to minimize crew time, which is at a premium. So we could conduct daily health checks to monitor animal welfare without taking up extra crew time. We wanted to perform multiple missions, capture some of what the shuttle was able to do for us in that era. The current plan for flying these is to conduct 2 flights per year. And finally, we wanted to make sure we had the science capability to apply cutting-edge technologies to any samples that we recover on orbit or after return, should those experiments be conducted. What do I mean by technical advances? Well, here are a couple of examples. One is genetically modified animals. These have already been applied in previous platforms, but there's a lot more work that needs to be done. What do I mean by genetically modified? Mice can be the gene sequence can be modified so that a particular gene of interest is overexpressed or underexpressed or knocked out. And that allows us to determine what the mechanism, how important that particular gene and gene product is for a given response. And a good example of that for a flight experiment is the Rodent Research 1 casus novartis experiment, which I'll talk about in a moment. Second new technology that we wanted to make sure our samples were good for was to be able to apply really cutting-edge technologies that have expanded so greatly in the last 10 to 15 years. And those are loosely referred to as omics. That is a characterization of a large pool of molecules. They may be genes, RNA transcripts, metabolites, that allows us to have greater insight into structure and function. And an example here from a previous flight experiment where this a technology was applied to learn something new and important is shown here. So, talking about challenges, what were our challenges in getting something to work here and getting it up and running? So we decided to adapt legacy hardware, taking advantage of the fact that we had 27 prior flights that successfully flew. We needed to interface that hardware with new vehicles. We don't have the shuttle to take anymore, so we worked with SpaceX and developed the capability to use the unmanned Dragon capsule to deliver the hardware. Finally, we needed to take care of the animals, provide husbandry. That entails training of crew and monitoring the animal welfare every day, which I already alluded to. This all seems pretty straightforward, doesn't it? Well, I'm going to delve into that for just a moment. This is the basic equipment that you need to conduct a rodent experiment on Earth. This is a standard mouse cage blown up here. The scientist can sit in the chair and observe the animals. And one thing, if you look at this, you can see, in fact, that, you know, this is pretty straightforward with gravity, how this works. The animal's in the cage, and, you know, the water comes down, and the waste falls into the bottom of the cage, and you provide this orange thing, in case you're curious, is enrichment. The animal plays and nests in this type of material. This is what you need to conduct an experiment on orbit. Not scaled to size. You need habitats that will manage the waste and provide the food and water in a way that is not gravity dependent. You need to protect the cabin environment, and so you need systems in which you handle the animals and transfer the animals in a controlled way. And you need a variety of kits and things to do all that in a safe and productive way. So let's talk about the system that was developed to accomplish that. There are two hardwares that the animals live in, the transport and the habitat. They look pretty similar on the outside. So I'm showing you this in a cutaway mode. The animals live inside here. And this is a port that allows you to access two chambers, both sides. To get into, there's an airflow system through here that captures the waste. There's also an animal access unit, which is a simple glove box type of arrangement that mates with either the transporter or the habitat so that the crew can go in, reach in, and recover the animals, placing them in a mouse transfer box, because you can't take an animal and walk across the room with it. It needs always to be contained, and it can be transferred in the mouse transfer box. And then we have too many kits to mention. This shows a view from the inside of where the animals live. There are Rodent Research 1, there were 5 animals per compartment, 2 compartments per hardware system. You'll see grating on all sides that allows the animals to ambulate, airflow and trap to be collected in filters. Food, these are food bars that are supplied continuously to the animal. There's a water supply that is not gravity dependent for delivery, which is, you can't see from this picture. Lighting, we supply a lights on, lights off cycle. The animals are most active in the dark cycle, as you may already know. And video cameras with infrared capability that allow us to monitor and observe the behavior of the animals. So let's develop our concept of operations here. How are we going to get them up there and get our samples? The animals are put in a transporter and delivered to Dragon, or mounted in Dragon as a late payload. Undergoes launch, can be, in the case of Rodent Research 1, it was 4 days in transit before docking, and the crew had, it was scheduled and conducted the transfer operation. The animal access unit was attached, the animals recovered and placed into habitats where they lived for for as long as 33 days, making the longest stay in microgravity 37 days. At the termination of the experiment, the animals were then transferred into the microgravity science glove box, which had been prepared, and the animals were humanely euthanized, and then tissues were retrieved and recovered under specific conditions that made sample analysis optimal. The samples were stowed and returned to Earth. So that's the plan, but we don't get to the start yet because we have to make sure it all works before we go. So we conducted extensive pre-flight testing to show that the animals thrive in the hardware, the operations work, and the samples recovered, as the crew would eventually do, would be done so in a way that got the expected science outcome. So now all systems go. Rodent Research 1. There were two main aspects, main objectives of Rodent Research 1. One was validation. The goal of this was to demonstrate the capability to support the health of the animals in long-duration experiments. This was achieved by evaluating all the key factors, including animal health, behavior, and tissue results, which I'll be talking about in a moment. In addition, there were mice flown for the National Lab, which CASIS, the Center for the Advancement of Science and Space, manages, and a Novartis scientist planned this experiment where MERF1 knockout mice, these again are mice that have a key gene knocked out and are resistant to muscle wasting, as well as control mice were flown. Here was our plan for sample retrieval. For validation, we were able to, CASIS kindly shared samples with us so we could work together to achieve our objectives. We recovered spleen, liver, also preserved animals for measuring body weight after return to Earth, and also for conducting post-flight tissue retrieval. So let me take a few minutes first, now as we start talking about how the experiment went, to talk about behavioral observations. Here are some qualitative observations that were made. When animals first entered the habitat, they very actively explored the compartments, much like they do routinely on Earth when you transfer them from one cage to a novel cage. They are also observed eating, drinking, and grooming. They groom both themselves and others while in the habitats, and these are all, again, considered normal behaviors of healthy mice. They were interesting to watch. Mice propelled themselves around the compartment in more than one way, mostly by pulling along the cage with their forelimbs, although their hind limbs were used to a more limited extent, also by floating from one location to another, and remarkably resembling how crew ambulate around the cabin. As time went on, the mice moved more and more quickly around the compartment. They translated with ease through the open spaces, but they also most often anchored themselves using their tails and paws. A detailed behavioral analysis is now being in progress. So I'm going to talk to you about our initial results with the tissues. First, I want to take a moment to explain the groups. There were, in fact, 4 different groups to evaluate the responses from the validation aspect of this flight. There was the spaceflight group, and there were 3 controls. So as I pointed out to you, changes occur rapidly over time with these animals because their lifespan is relatively short. So one independent variable in this design of a long-duration experiment is time. What are the changes that take place over time just due to aging? And so for that, we have what we call a basal group. These animals were euthanized and tissues recovered at the time of launch from the same group of animals that the flight animals came from. They were you can compare those results to those of animals that were maintained in standard cages the way we normally do and where an investigator in a lab anywhere would do, and answer the question, what is the effect of time as an independent variable on a given outcome? What's the other variable here? The other variable is cage. So the habitat, as you can imagine, nobody does experiments in those habitats unless you're planning to go to space. And in fact, changes in the environment of the animals can have very profound consequences for basic physiological and cellular responses. And so we, to facilitate future investigators' ability to evaluate whether the changes in their control groups are due to the cage, we evaluate and compare this vivarium group in standard cages to the ground controls. And the ground controls are the group of mice that are housed in identical cages to the flight. They're also kept in an environmental chamber at Kennedy Space Center that has the environment of carbon dioxide, temperature, and humidity matched to ambient conditions in the cabin on the space station, because we want the main variable in making the comparison between spaceflight and ground control animals to be space. And that is the final and key comparison. So this summarizes the results we've obtained to date. We have body weight and tissue weights from the validation mice. I'm not going to talk about the variable of time and cage in detail, but fortunately there were not huge differences, which yields a simpler analysis. Now we can, in the case of the spaceflight environment, we can simply compare the ground controls to the flight group and ask what were the direction of changes. Now, one thing I've done here is I've put in green the changes or lack of changes that is new information. These are not changes that we observed in shorter duration shuttle missions. They are different in direction from some of those experiments. And so going through them sequentially, we saw no difference in body weights between the ground control and the flight, nor compared to any of the other groups. There was an increase in liver mass. There was no effect on adrenal gland. Now, the adrenal gland is responsible for producing a principal stress hormone in the body. In humans, it's cortisol. In mice, it's corticosterone. And previous short duration experiments on occasion observed hypertrophy or growth of the adrenal gland, which can occur in response to a chronic stressor. So there was no effect of the space environment on the gland size. The thymus and the spleen are two glands that are involved in the immune response. We saw an increase in mass. Shuttle missions in mice have shown a decrease in short duration, and a decrease in spleen mass. Now, what we did see that was consistent with previous missions is a decline in the soleus muscle mass. And the soleus muscle is an anti-gravity or postural muscle in the leg that atrophies in response to disuse or microgravity. And that was a very consistent finding. What about the quality of our sample retrieved? So we evaluated RNA quality recovered from the liver and the spleen, and looked at the quantitative value of quality. And we found from the flight animals as well as the ground controls that the quality was acceptable for even the most demanding of analyses. RNA-seq is a method that allows it to sequence all of the transcripts, RNA transcripts in a sample, and is demanding for high RNA quality. In fact, we've achieved that. The analysis hasn't been complete, nor has the liver enzyme activity analyses that are still in progress. But we achieved our goal of obtaining samples of adequate quality for applying these very demanding techniques. Now, how can we expand our science outcome from our original goals? One way we do that is through biospecimen sharing. So when we got the samples back from the station The project recovered 32 tissues from 40 validation mice, which yielded more than 3,000 vials of tissues that are now being stored in the Life Sciences Data Archive. These are destined for distribution through the biospecimen sharing program of space biology, which includes our Russian colleagues at IMBP who've requested tissues. In addition, some of the tissues will go to the NASA Gene Lab project, which I'll talk about in a moment. I just want to take one moment to describe the biospecimen sharing because it's been so successful in the past. The images show a team that traveled to Russia to recover samples from the Bion M1 mission. And you can see from the outcome from previous flights, this is only a select group of scientific manuscripts that came out of a single shuttle experiment, how insight into multiple systems can be derived from this type of approach. So I'm very excited about the future for the samples coming out of Rodent Research 1. In addition, we provided liver samples to the Gene Lab project who are processing these samples to analyze the RNA transcripts, DNA modifications, and protein profile in the samples in some detail. And these samples and data sets will be made available to the scientific community. So I'm going to take a moment to summarize now where we are so far with rodent research. The hardware and operations were performed successfully on orbit. We got it all the way through to sample return. The mice thrived through 37 days in microgravity. Some important analyses are still in progress. It's important to note that common indicators of stress were not observed in the animals, such as a loss of body weight, an increase in adrenal gland weight. They were the same in all the groups. Also, the preliminary findings on wet tissue masses contrasted sharply to findings from shuttle experiments, which were shorter in duration, also had other variables, but duration may be one of the key variables in defining those differences. And with biospecimen sharing, much more to come. So I would suggest from these findings, and this is a hypothesis, not a conclusion, that there are at least two phases of physiological changes that occur after entry into the spaceflight environment. Of course, we don't know yet how far they will progress. So I'm addressing the audience to think about and invite you to pose your own hypotheses that might explain such a thing. But one thing I'd like you to keep in mind as you think about this in the future, this sort of diagrammatically represents the problem that's being posed with the magnitude of the response on the y-axis and time in space on the x-axis. Each of the colors represents a different organ system response. So the green may represent the vestibular response to microgravity, which occurs very rapidly. The red may be cardiovascular adaptations, which improve over time, and the blue may be another system that takes a longer time. So these various time dependencies need to be taken into account as you think about whether or not duration in space is a key variable and might explain our results and help us design new spaceflight experiments. Maybe we'll get some insight from Rodent Research 2. That mission, the on-orbit activities have been completed for Rodent Research 2. That's a CASIS mission that lasted 60 days. So perhaps we'll get insight into that. So I'd like to wrap up now with a couple concluding comments. To live in space, hopefully I've effectively shared with you that there are multiple challenges to the human body that are posed by that environment, and that many different physiological systems can be affected. And I believe the resulting complexity is such that the consequences of these challenges and these responses are such that it's virtually impossible to predict with certainty what those consequences will be for human health, for human reproduction over very long periods of time. We believe that insight into those responses and mechanisms will improve our ability to predict and potentially mitigate, make decisions about what we need to do to protect humans in space, living in space, and also that rodent research on the ISS will help us get there. I'll finish up with some acknowledgments. This shows a very happy group of people after staying up all night, talking the astronaut crew through some difficult on-orbit operations in Rodent Research 1. But of course, they are a very small sample of the number of people who actually had a hand in making rodent research work on station. It would vie with the number of people that showed up to break the Guinness Book of World Records for dancing at the same place at the same time in Mexico City, but still there are many important people who can't be named here. And also I want to mention the inspiration that comes from the research lab that I work in, the Bone and Signaling Lab. And I'm going to finish up here and leave up during the Q&A period. First, thank you very, very much for listening so patiently. And also, I'm going to leave this up in case you have interest in following up on with some of these links and some aspects of what I've talked about today.
So thank you, Ruth. We have a few minutes for questions. If you have a question, please line up on the microphone in the center of the aisle. Please be brief and no follow-up questions. Thank you.
Ruth, is this the first time that mice have been in space for that 30-day duration? Is that the longest, one of the longest periods? And my follow-on question, sorry, I'll ask, is, basically I'm just really fascinated about what would happen with adaptation of those animals over that long period of time in terms of behavior, interacting with feed systems, interacting with each other, that kind of thing.
Right, well, so there were two other flight experiments that one 30 days. The Bion mission was a 30-day mission, and the mouse drawer system experiment was a 90-day mission that was flown once. You pose a good question about interacting over time, and that is one of the reasons why we are performing a very careful and quantitative behavioral analysis of the video collected from that mission. But I think we need more longer duration missions so we can capture a larger fraction of that total time and space.
Hi. I was wondering if there were any plans to do partial gravity simulations in space or on the ground, like 1/3.
So JAXA, the Japanese Space Agency, is is working on flying an rodent experiment now, where I do believe the plan is to completely replace the gravity vector. But once the capability is developed for centrifugation, then it's possible to do partial gravity experiments, and there's a lot of interest in doing that, in thinking about going to other planets.
Would it be helpful to have a centrifuge so you could tease out the effects of weightlessness versus radiation?
Absolutely. It would be great.
Ruth, there's more data on humans in space in terms of duration than there are on mice. Have you done anything to compare the mice results to data you've gotten from humans?
That's a really good question, because fundamentally, if we want to extrapolate from the result, the assumption of these studies is that they will yield insight into humans. So that's a work, as you can imagine, the answer to that question is a work in progress. There are limits to every model system, and as in terms of going forward with the new capability we have, that's going to be an important aspect to look at. So, a work in progress.
So one follow-on question, and that is, do you have a data that suggest where an endpoint would be? I mean, we know the endocrine system is damaged, the muscles are problematic, the skeleton has issues. At what point would we decide, well, gee, we can't do space. It's going to be too difficult. We're never going to be able to colonize space. Do you have a feeling after 40 years of data collection?
Well, I am not sure that I would answer that question. That is a question that's going to take more than rodent experiments to answer. It's an excellent question without a single endpoint, and I'm sorry I don't have a better answer for you. But I think that the answer to that question needs to take into consideration both our ability to extrapolate from the rodent experiments, also an understanding of when the changes that we observe pass the bar from subclinical into clinical significance. And that's very important. The Human Research Program at NASA spends a lot of time reviewing that. And they've compiled evidence books in all, related to all of the major identified risks to human health. And part of that process, the goal of that process, is really to identify when do you see a change that's acceptable and when do you pass into significant versus very severe risk. And so I would say there's a whole program devoted to answering that question.
All right, please join me in thanking Dr. Ruth Globus for an excellent seminar.